Hi everyone, so we are now in Module 5 material, which is our last module of the semester. And in this topic, we are going to talk about protein synthesis, which means how proteins are made. Our last section was on genetics, and we talked about how genes are passed on from one generation to the next, and that's how you inherit traits. And that's one function of the DNA in our cells, is that it provides the instructions for making proteins that we pass on to our offspring. So now we're going to talk a little bit more about how those genes function on a day-to-day basis of giving the instructions for making proteins. So if you go back, this would be a cell, and inside of the nucleus, you have your DNA, which is organized in chromosomes, which we just finished talking about. Chromosomes are what we inherit from our parents, a set of chromosomes from each parent. And the chromosomes are made up of DNA. And the DNA in a chromosome is highly coiled so that this chromosome doesn't represent one piece, but a highly twisted piece of DNA. So if you could stretch out the DNA in one of your cells, it would be about 3 feet long. So in the chromosome, the DNA is wrapped around a protein, so it kind of gives it a base for its structure called a histone. And if you were to untwist one of those strands in the chromosome, a section of that DNA would be a gene, and that gene is composed of individual components of DNA. So DNA is a very big polymer. Remember, we talked early in the semester about polymers, and polymers are large molecules that are made up of monomers. And monomers are kind of the repeating unit. Remember, we talked about starch is a polymer and the monomer is glucose. So when you make starch, you have thousands of glucoses attached to each other. Proteins are polymers made up of amino acids. DNA is a polymer, and it's made up of nucleotides. So we're going to talk first a little bit more specifically about the structure of DNA, and then we'll talk about how it's used to make proteins and what happens when there is a mutation in the DNA and you don't make the protein correctly. So that's what we'll be talking about in this section. So again, chromosomes are made up of DNA. They're very long strands of DNA. And the monomer in DNA is called a nucleotide. And we talked about that all humans, in all of our cells, except for sperm and egg cells, we have 46 chromosomes. So each of those chromosomes are these coiled strands of DNA. And they contain specific genes. Generally speaking, a gene represents the instructions for making one of our proteins. And we have thousands of different proteins. We have about 25,000 genes that are scattered across those 46 chromosomes. It's actually 23 because we have 2 copies of every gene on the 46 chromosomes. So a single chromosome has many genes. We have 25,000 genes, only 23 chromosomes. So each chromosome has to have hundreds or thousands of genes on them. All of your genes together is called your genome. So your genome is figuring out what all genes you have and what the composition of those genes are. And they have figured out the human genome. They have sequenced the human genome. So you can go online and Google human genome, but our DNA is billions of nucleotides. So it has a list of what all those nucleotides are. I just was reading an article that the coronavirus, they've done its genome, and it's much, much, much smaller than the human genome. And they know what all the genes are, and they think they know mostly what each of those genes represent. So chromosome is the more complex, the big polymer structure, which is composed of genes. which is made up of nucleotides of DNA. Excuse me. So this is looking at that structure of DNA in a little more detail. So, and if we had done the DNA lab in person rather than virtually, you would have extracted your own DNA from your cheek cells and been able to take it home with you if you so desired. So DNA is called a double helix, and it's a twisted double helix. So this is what it looks like. It's kind of like if you had a ladder and you twisted it. That's the structure of DNA. And each DNA, it's a double helix, so that means it has two sides. So this is like one side, and then this is the other side. And they're joined together in the middle. It's made up of repeating units that are called nucleotides. So remember, that's the monomer for DNA, is a nucleotide. So your DNA in your cell has billions of nucleotides. So this would be one nucleotide. They have it in color here. And each nucleotide, and we'll talk about this more, has three parts. It has a phosphate group, a sugar, and a base. So it's a phosphate, sugar, and a base. And so that's one nucleotide. This is the next nucleotide. And then going on the other direction, here's a nucleotide, and here's a nucleotide. So this is just a little tiny piece of DNA, but the sugar and the bases kind of form the rails of the ladder. I mean, the sugar and the phosphates. And then the bases are the middle part. Like if you were climbing a ladder, it would be the part that you stepped on. So this is kind of a blown up picture of a piece of DNA. And you can see, again, this would be one nucleotide. So each nucleotide is a phosphate group, which is here, a sugar, which is here. The sugar in DNA is deoxyribose, and that's why it gets its name of DNA. So it's deoxyribose nucleic acid. That's the sugar, and then this is the base. And then this phosphate is hooked to the sugar of the next nucleotide. So then you have phosphate, sugar, base, phosphate, sugar, base, phosphate, sugar, base, phosphate, sugar, base, on and on and on, billions of times in your nucleus of each cell. And then on the other side, it's the same thing. Phosphate, sugar, base, phosphate, sugar, base, phosphate, sugar, base, phosphate, sugar, base, phosphate, sugar, base. And then the bases join together in the middle. So along the side, we have phosphate attached to sugar. That's a covalent bond. Remember we talked about covalent bonds where molecules share electrons? So those are covalent bonds. The hydrogen bonds are in the middle. We talked about hydrogen bonds with water, where H plus is attracted to an O minus. So that's what holds the DNA together. Now, again, each nucleotide, I think I've said this a couple times, is composed of a base, a sugar, and a phosphate. The phosphate's made from DNA, ATP, so one of those phosphates is from ATP. So there are 4 different bases in DNA, and usually they're just referred to by the letter. And the 4 bases are cytosine, thymine. This one is not in DNA, it's in RNA. We'll talk about that separate. Adenine and guanine. And they're usually referred to as A, G, C, and T. So those are the 4 bases that make up the bases of DNA that are attached to the sugar and kind of stick in the middle of the ladder. So again, these are the 4 bases: adenine, thymine, guanine, and cytosine. So A, T, G, C. And so for every nucleotide, you have a phosphate, a sugar, and a base. A phosphate, a sugar, and a base. And that's the repeating unit of DNA to make up the polymer, a phosphate, sugar, and base. And the base is going to be one of these four bases, either A, T, G, or C. The phosphate in the sugar is the same for every nucleotide. The only part that's different are the four bases are different, either A, T, G, or C. You can just remember those. This is a video which it should work if you want to watch it. It's kind of fun, but that's if it works on your computer, you can watch it. So again, the DNA is a twisted double helix ladder, and the rails, which are like the side pieces, are also sometimes called the backbone. And that's phosphate and sugar. Phosphate, sugar, phosphate, sugar, phosphate, sugar. Oops. And then the middle part are the two bases. And then it's phosphate, sugar, phosphate, sugar. I'm not a very good artist. The way the bases work is that the only way it all fits together is that A is always attached to T. It could be on either side, so you could have A here and T here, or you could have T here and A here, or G is attached to C. So you could have G, oops, C, or G, C. You never have A attached to C or T attached to C or G. It's always AT and GC. One side of the DNA, so if you were to, it's called unzip it and have just one of the strands, is called a template strand, and that's what's used to make proteins. The other side is the non-template strand, and it's important in genetics, but it's not used to make proteins. So since we're talking primarily about the protein making function of DNA today, we won't really be talking about the non-template strand. So the way that our cells use DNA to control the activities of the cell is by using the genes to make specific proteins that we need. So if you need more insulin, you have a gene for insulin which can get activated and result in more insulin protein being made to meet your needs for insulin. If you need more hemoglobin, if you need more of an enzyme, you have a gene for all of those proteins that you can, it's called turn on or turn off, but you can activate it to make the protein or unactivate it so it's not making any protein. So the process involved in making proteins from the DNA, the information in the gene, involves two processes. One is called transcription. And with transcription, you have your DNA inside of your nucleus. And as I said, it's a really big molecule. If you remember from when we talked about the functions of all the cell parts, we said that ribosomes are where proteins are made. So the ribosomes are out in the cytoplasm. DNA is this giant molecule inside the nucleus. So DNA has the instructions for making a protein. You can think of DNA like a cookbook. It has the recipes for making your thousands of different proteins you make. But they're not going to be made inside the nucleus. They're made out in the ribosome. So you need a way of getting those instructions from the DNA in the nucleus out to the ribosome. And the way that we do that is we use a molecule related to DNA but slightly different called RNA. So the first part of making a protein is going from the DNA to the RNA so that the RNA copy of the gene is made, and that's called transcription. Then that RNA leaves the nucleus, goes out, attaches to the ribosome, and makes the protein. and that is called translation. We're going to be talking about each of these in a lot of detail to make it a little bit clearer. Excuse me. So again, this is showing the same thing. So here we have our double-stranded DNA sitting inside of the nucleus. And suppose we need a specific gene. We need to make more copies of insulin. We'll stick with insulin because most of you are familiar with insulin and know what it does. So we'll take the gene for insulin, we unzip it, which means we untwist it, and so that the bases aren't hooked together, and we use the sequence for the DNA on the template strand to make a copy in the form of RNA. And so that is the process of transcription, is making the RNA that complements the gene in the nucleus. Then that gets processed, leaves the nucleus, goes out into the cytoplasm. It attaches to the ribosome, which is this peachy kind of thing, and you make your protein, and that's called translation. Before we go into the details about that process, I wanted to just talk a little bit more about RNA. So RNA, as you can kind of get from the name, it's similar. So you have DNA and RNA. And DNA stands for ribonucleic acid, and RNA is ribonucleic acid. So RNA structure, very similar to DNA, has a couple important differences. One is that it still has 4 bases, and it has A, G, and C, but it doesn't have T. Instead of T, it has uracil. So instead of a T, it has a U. And so U would attach to A instead of T. Also, it's single-stranded, which means it only is one half of the ladder. It doesn't form the double helix. And the third difference is that the sugar is not deoxyribose, which is the sugar in the nucleotides in DNA, but it's ribose, which is very similar. So it's ribonucleic acid. So it still has nucleotides that have a phosphate, a sugar, and a base. The sugar is different, and the one of the bases is different. There are 3 different forms of RNA, but I'm not going to talk about them now. I'll talk about them as we go through the process and how the 3 different forms are used. And I think it'll be less confusing if we do it that way. So here is a little piece of RNA. And again, it's made up of nucleotides, just like DNA, has a phosphate group, a sugar, and a base. Phosphate, sugar, base, phosphate, sugar, base, phosphate, sugar, base. But there's not another side, so that base isn't attached to another base to form a double helix. And there are no T's. There are U's instead of T's. And again, the sugar is a little bit different. It's ribose. But it's still quite similar. It's a polymer made up of nucleotides, all of which have a phosphate, sugar, and a base. So in the part of the first process of making a protein is taking your gene. So again, say we have the gene for insulin and we need to make more insulin. We just ate a soda and 3 cookies and a cup of ice cream. So we need more insulin to get rid of all that sugar. So this would say the gene for insulin. We go through transcription where we make RNA that's complementary to that gene for insulin. And that's the process of transcription. That RNA leaves the nucleus, goes out to the cytoplasm where we would make the protein insulin using the instructions from RNA, which actually came from DNA. So how are these instructions used? Like, how do we know? Remember, protein is a polymer of amino acids. And you may remember that there are 20 different amino acids. And what is different from one protein to the next is which amino acids are used to make the protein. So a gene, what it does is it gives the instructions for putting together a protein by saying, first you take this amino acid, and then the next amino acid, and then the next one until you're all done. Insulin has 31 amino acids, so it's instructions for which of those amino acids and in what order they go, the 31 amino acids to make insulin. So the way that the DNA does that is it uses something called codons. And a codon are every 3 bases in the gene of the DNA. And the bases are used by the letters. So say you had a codon would be ATC or A or GAC. Those would be codons. There are 3 bases and they represent a particular amino acid. So that is the process used for making our proteins. The kind of the instructions is called the genetic code of which 3 bases represents which amino acid. That code is called universal, and what that means, we don't really know if it's everywhere in the universe, but everywhere on Earth, the same codons are used by all living organisms. So whatever amino acid ATC represents, it's the same whether you're an E. coli or whether you're a fish or whether you're a flower or whether you're a human, it represents the same amino acids. That fact is why you can put genes for different organisms. Like we actually can make insulin in bacteria by putting the human gene in bacteria, and it will use the same code for making the protein as we do and come up with human insulin. So we're going to talk more about how that's done. There are also codons that say this is the beginning, which are called the start codons, and then the terminator codons that say this is where this protein ends. So all these provide the instruction for making a particular protein. So here is your double-stranded DNA, just a little tiny piece, and then the template strand uses that sequence to make RNA. So here the template strand is this bottom one here. So we have the first codon is TCG. So when you make the RNA, it's the complementary basis. So T goes with A, C with G, G with, that should be a C here. I can't tell if it is or not. And then that codon in your RNA, AGC, represents a specific amino acid. So that's how your cell knows to put that amino acid into that protein, is by the codons that are in the DNA and then in the RNA. So we're going to go into more details about this. I had to split the PowerPoint in half, so it will continue in protein synthesis part 2.